We ended last class talking about DNA and where it's located within the nucleus. Well, now we get to start looking at how we create a DNA, or excuse me, a protein. So we're getting into protein synthesis. And so when we look at the functions of the nucleus and the ribosomes, they are actually involved with protein synthesis. Cellular activities are dependent upon this particular process. And this process of protein synthesis is directed by the DNA molecule that's located within the nucleus. Now, there are a couple steps or a couple phases of protein synthesis. The first one is transcription. And transcription involves RNA. And RNA, in particular, this messenger RNA or mRNA, is a copy of a gene that's formed from the DNA in the nucleus. Our second process is translation. And translation takes that mRNA molecule and uses it for the synthesis of the protein by the ribosomes in the cytosol. So we'll actually talk about both of those here in just a second. I am going to type this here right beside it. When you hear me talk about it, it's called mRNA, okay? And this is our messenger RNA molecule. And so we have a couple images over here that basically kind of show in graphical representation kind of a basic overview of it, but we'll go into detail of those. So in order to synthesize RNA, this is the process known as transcription. So we're basically transcribing an RNA molecule. So there are a couple processes or a couple structures that are required. DNA is the major structure that's required in this process of transcription. And so what DNA does is it's going to serve as a template for a complementary RNA molecule. So again, if we remember what RNA is this repeating series of ribonucleotides, okay? So we have these ribonucleotide monomers that basically form one right after the other. In DNA, it was A, T, C, and G. In RNA, it's A, U, C, and G. So we end up having, again, a ribose sugar, a phosphate, and one of the four nitrogenous bases of, again, A, U, C, and G. Now, with DNA, we have a double strand. With RNA, we have a single strand of these nucleotides. Other structures, we need to have a large number of the ribonucleotides, as well as this particular enzyme located here at the bottom, and that's called RNA polymerase. And RNA polymerase is going to be located within the nucleoplasm in the nucleus. And this is where we're actually going to transcribe our RNA molecule from our DNA molecule. So there are three types of RNA produced during transcription. We have messenger RNA, we have transfer RNA, and we have ribosomal RNA, also known as mRNA, tRNA, and rRNA. We have three events of transcription. We have initiation, elongation, and termination. And those phases or those events are exactly like they sound. We initiate the process, we are elongating our mRNA molecule, and then we terminate the creation of that molecule. So let's look at these phases. So we have first initiation. So in this case, we have the DNA molecule is unwound by specific enzymes to make it accessible to the RNA polymerase. And again, RNA polymerase is the enzyme that catalyzes the synthesis of mRNA. So we then have the the RNA polymerase molecule comes in and attaches to the DNA strand, and it locates what we call a promoter region. And this promoter region, I'll just kind of circle them here. This promoter region serves as the start point for gene transcription. And it's at this point where we're now breaking bonds between the DNA, the two strands of the DNA molecule. Remember, if you think about DNA and it's that double helix structure, those DNA nucleotides are actually held together. The two strands are held together by hydrogen bonds. And so we go in and we break those hydrogen bonds, and now we have those DNA nucleotides of one strand exposed. So now we separate it, and the region that the RNA polymerase molecule is located now starts to expand. And it's at this point that we start to copy our DNA. So the DNA strand that's actually being copied is called the template strand. The other strand that's not being copied by the RNA polymerase is called the coding strand. Now, our second phase in transcription is elongation. And elongation is where we're now taking those RNA nucleotides and adding them to our mRNA molecule. And so we're elongating this particular mRNA strand. So we have these free ribonucleotides are base paired, and they pair with the exposed bases on the DNA template. And the pairing is basically the same as what we've already learned. As in DNA, A and T go together, but with this, now we have A goes with U. And on the DNA molecule, when we have a T, that now pairs with A on the RNA molecule. We're also forming hydrogen bonds between the ribonucleotide and the complementary DNA strand. So it's almost as if we're forming a kind of a 1.5 strands, if you will. We're now forming these new phosphodiester bonds between the ribonucleotides, and this helps to form our RNA polymer. Again, this structure of multiple monomers, the monomers being those ribonucleotides. Our RNA polymerase continues to slide down and move down the DNA molecule, and it continues this process until the entire gene is transcribed. When we get to termination, then, termination is obviously the stop point. This is where the RNA polymerase reaches a terminal region of the gene and is released. At this point, our hydrogen bonds are broken between the DNA and our new mRNA strand that we just created. And our DNA then rewinds back into its double helix, and now we have our double-stranded DNA molecule back together, and we have our new mRNA strand by itself. So again, hopefully you are able to see this okay on your computer screens or your tablets, but this is our graphical representation of transcription. So again, if we take a look here, up where my highlighted pointer is, this is our DNA molecule. So we now have to take this DNA molecule and unwind the two strands. This is done by our RNA polymerase molecule, okay, or RNA polymerase enzyme. Now in this process, you'll want to note that RNA polymerase functionally does pretty much everything. One, it attaches to the DNA strand, it unwinds the DNA strand, it adds our complementary RNA base pairs, and then it rewinds the DNA strand. So all of those functions are all part of RNA polymerase. So we have the initiation process where the DNA is unwound by the enzymes, particularly with RNA polymerase. And so as it attaches there to this unwound or opened up strand, it'll actually start to read the DNA molecule. Again, note there are two strands of DNA. We have our coding strand here. We have our template strand here. The coding strand is going to be the side that's not being transcribed. The template strand is going to be the part that is being transcribed. Here we represent a promoter region, which basically is going to represent the start point. So this is the initiation phase as we read that promoter region. Again, now we go through elongation, and so now we're adding complementary base pairs. So as our RNA polymerase molecule continues to move in this direction, it continues to read this template strand. And as it reads the template strand, it begins to add more base pairs. So we add the base pair, it adds A, U, C, and G to create a longer RNA molecule until we get to the ending point or the termination signal. And on here, you'll see it represented further down the strand. So as this RNA molecule moves in this right to left direction, it's continuing to read this DNA molecule until we get to now this terminal region. You'll see down here, we reach the terminal region. Our RNA polymerase basically pops off and has now rewound our DNA molecule and has now created this strand of mRNA. Now you'll notice that we call this a pre-mRNA strand, and you'll notice it's indicated here with multiple levels of introns and exons, and we'll talk about those in just a second. So after we've created or transcribed this mRNA molecule from DNA, we go through some modifications. And so initially, that synthesized mRNA molecule is more specifically called a pre-mRNA molecule because it's actually going to undergo some changes before it leaves the nucleus. After we go through some of these modifications, where we take out pieces and we splice them back together, this now allows us to form a mature mRNA molecule. And this mRNA molecule is now going to be used as the recipe to make or create a protein. So as we go through this splicing process, some of the non-coding regions of the pre-mRNA are called introns. And introns are actually pulled and removed from the pre-mRNA molecule. What we're left with then are these exons. And these exons are then subsequently spliced back together so that essentially we just have a full strand of exons. And these exons are then transcribed or are a transcription of a nucleotide sequence in the mRNA for a protein. So that was transcription. So our next phase in our protein synthesis is called translation. And so translation basically is what it sounds like. We are going to take that mRNA molecule that we just transcribed, and we're now going to translate it from mRNA language. We're going to translate it into protein language. And so this is essentially the synthesis of a new protein. And it's going to occur at the ribosomes within the cytoplasm and around the endoplasmic reticulum. The mRNA is actually threaded through a ribosome, kind of like threading a piece of thread through the eye of a needle. And the code in the nucleotide sequence of the mRNA molecule is now translated. And this code is actually converted from our mRNA language of A, U, C, and Gs into amino acids that we use to help produce a protein. So some of the required structures in translation. These are going to include ribosomes, our mRNA molecule, tRNA molecules, and amino acids. From these four structures, then, a protein is the product that we're actually looking to form. So let's look at ribosomes first. So ribosomes are actually composed of our RNA and protein. We do have a couple subunits. We have a large ribosomal subunit and a small ribosomal subunit. Remember, we talked about this before, that on our large subunit of our ribosome, we're going to have an A site, a P site, and an E site. Okay. Now, our rRNAs actually serve as catalysts during the assembly of the amino acids into a protein molecule. So it does help speed up that process. We already know that our mRNA is this structure of RNA that's transcribed from our genes. We know that it carries the instructions or the blueprints for how we're going to synthesize particular proteins. And again, knowing what it is, it's a linear sequence of these nucleotides. So when we look at the mRNA molecule and how it's actually threaded into the ribosome, we look at this long sequence of amino acid, or excuse me, of nucleotides, and how it's arranged in numerous A, U, C's, and G's. And so as our ribosome starts to read our mRNA molecule, it's actually read 3 base pairs at a time. So we're actually reading it in 3-letter sequences, okay? And so we call this a codon. And so a codon is a 3-base unit. Now there are numerous types of codons. Particularly, we have a start codon. Now a start codon contains the nucleotides AUG. And this start codon basically signals to begin protein synthesis. Everything after that are basically just base pairs or codons arranged in these 3-base units. So the codons following start and the stop aid in the direct assembly. Now we also then have the stop codon. And so the stop codon is where the mRNA molecule basically stops the reading, or the ribosome stops the reading of the mRNA molecule, okay? And so we start the process with a start codon, which is going to be AUG. We have this series of additional codons that follow the start codon, but occur before the stop codon. And this, again, is this direct assembly of our protein. And then we reach the stop codon. And this is where the ribosome stops reading that mRNA molecule. Our tRNA then is the RNA component that actually brings the specific amino acids to a specific rRNA codon. This gives us kind of a, or looks as if it's kind of a clover leaf shape, but it does contain an amino acid on the end of it. So it has an amino acid acceptor region where it provides the attachment site for specific amino acids. Within that tRNA molecule is called an anticodon region. And so an anticodon region is also a sequence of 3 nucleotides that is read, and it determines the specific amino acid to which the tRNA attaches. It is catalyzed by amino-acyl tRNA synthetase, and after binding of the amino acid, the tRNA is what we call a charged tRNA molecule. And so a charged tRNA molecule does contain an amino acid on it. And so it's going to serve as the adapter site for binding the tRNA to a complementary codon of the mRNA. So again, what we're going to see is that on the mRNA molecule, we have these 3 nucleotide bases. That's called a codon. The tRNA is going to have the opposite 3-base sequence, and it's going to bring in the complementary amino acid. We do have 20 amino acids that are found in proteins of living things. And so here we take a look at kind of these structures of what we're utilizing. So here we have our ribosome. We have the large subunit and the small subunit. Again, we can indicate the E site, the P site, and the A site. Okay. Here we have our messenger RNA. Okay. And as we read this, again, we're looking at every 3 nucleotides. So here's AUG. So this is going to be our start codon. This indicates the starting point of our protein. We then have an additional series of 3, an additional series of 3, so on and so forth, until we reach, in this case, UAA, which is going to indicate the stop codon. Here's our cloverleaf tRNA molecule. And so we have the amino acid acceptor region on this end. And then we have the representation for tRNA as we contain the anticodon. Again, we have the amino acid acceptor end on this side. So this is kind of the cloverleaf version of it as far as what we look at from a very basic standpoint. This is more the representation of how we're going to indicate it. Here are some amino acids and protein building blocks. Here's a representation of an amino acid. Again, we have the amine side, kind of here in pink if you can see that. Here we have our carboxylic acid side, our C double bonded O to an O and then to an OH. And then we have our R region, and that R region can indicate a series of different groups. It can be as simple as a hydrogen atom that's bonded to a carbon there. It can represent a methyl group, which is going to be another carbon atom that has 3 hydrogen attached to it. It can contain sulfur groups, nitrogen groups. So there's a lot of different types of structures that can be contained in that R group. This is going to be the representation for the amino acid, just kind of a little bit of a bubble there. And here we have a representation of a strand of amino acids. If we remember going back to our structures of proteins, this strand of amino acids would represent primary structure of the protein. We then move into the protein, where in here you'll see our alpha helices and our beta pleated sheets. The alpha helices and the beta pleated sheets themselves indicate secondary structure. When we start folding them into more of a 3D conformation, we get tertiary structure. And then if we add multiple series of tertiary structures together, we end up with a functional protein, and that is our quaternary structure. So this is our genetic code, basically. We are looking at the differences or the series of nucleotides in 3 base sequences. So here you can see what they actually code for, different amino acids. Obviously, some amino acids are going to be coded by multiple sections of codons and anticodons, whereas some of them are going to be one, and that's what we see here with methionine, is indicated by the AUG, and it indicates the start codon. Now, we don't need to know all these. We don't need to memorize these. Obviously, this is just kind of a table to kind of give you some representation. But this is essentially the genetic code as we read DNA and into mRNA, and then bring in our tRNA molecules with our amino acids attached to them. So there are 3 events to translation. And these 3 events are how we're actually going to create or synthesize the protein, okay? So just as we had these 3 events in transcription as we created our mRNA molecule, we also have these events in the synthesizing of a protein. So again, we have initiation, elongation, and termination. So now with initiation, we have the complex that's formed between the ribosomal subunits, the mRNA, and the tRNA. So we have the base pairs between the tRNA and the start codon in the mRNA molecule. So methionine is going to be bound to this particular tRNA molecule as it codes with the anticodon on the tRNA. So methionine is always going to be the first amino acid in protein synthesis. However, it can later be removed and spliced off if the instructions call for that. And so now we have the start codon is now on the ribosomal P site. So it basically started in the A site where we attach it, and now as our ribosome starts to move down our mRNA molecule, it now gets shifted and moved into the P site. Elongation is just what it sounds like. We're elongating it or creating a longer protein. So now we're actually bringing in subsequent amino acids by specific tRNAs to form that particular protein. Now there are 3 steps with elongation. We have the charged tRNA with the complementary anticodon base pairs, and it comes in and binds to the codon of the mRNA in the A site. We form the peptide bond, which is now going to be between 2 separate amino acids. And this occurs while the tRNAs are in the P site and the A site. The ribosome then moves down 1 codon So it's kind of moving downstream. At this point, the tRNA that's in the A site moves to the P site. That was just in the P site then gets moved to the E site, where basically it's ejected. So now the A site opens up again, and the uncharged tRNA that released its amino acid is now released from the E site. And this process repeats until the entire mRNA sequence is translated. Okay? And as this occurs, it basically yields a linear strand of amino acids that we know as primary structure. We then have termination. And so termination is the entrance of the stop codon into the A site to end translation. It has a release factor that's going to enter that A site instead of some sort of charged tRNA molecule. And the two ribosomal units then separate as the new protein is then released. So this is kind of our process. And again, I apologize if it's relatively small. But this does kind of show us what we're actually looking at as far as the process of protein synthesis, okay? And so you may want to follow along in your books. Please understand that I do also have a video in Canvas that you can go and watch. I have linked some of these videos in YouTube that you can use as kind of a refresher or in a way to actually see it in process as it's actually occurring, as opposed to just seeing the steps visually on the screen here. So again, if we look at this process up here is, again, is transcription. And so we have our DNA that's being created by our RNA polymerase. We create a pre-mRNA molecule, it gets spliced, and we're left with our mRNA molecule. This mRNA molecule now begins to go through translation. And so we have our two ribosomal subunits, the large unit and the small unit, that bind to our mRNA molecule, or the mRNA molecule is threaded into it. So now our ribosome begins to read this mRNA molecule. We get that first sequence, that AUG sequence that you can see right where my pointer is. And so it now has the codon AUG. This is what codes for methionine. And so we also then have a tRNA molecule that comes through. So as our ribosome begins to read it, it hits the A site. We now bring in a tRNA molecule with an anticodon of UAC. Okay? So again, the anticodon is going to be the opposite of our codon. And so it attaches here at the P site. We now have an open A site where we're now able to read the next series of letters, which is going to be our next codon. It's going to code for a specific amino acid. We have a tRNA molecule that's charged, that has an amino acid on it, and has the opposite three-letter series, which again is our anticodon. Our tRNA molecule comes in, it binds at the A site. We have our first one in the P site, and now we're actually creating this peptide bond between the two amino acids. Our ribosome continues to move across, and so now we have an open A site again. Our first tRNA molecule got put into the E site, where it's now ejected and is uncharged with no amino acid attached to it. And now we're able to bring in a new tRNA molecule into the A site. We then form another peptide bond. That ribosome moves over. The uncharged tRNA is released, and we're left with that next tRNA molecule. And so this process continues until we reach termination. Okay? And so termination is where we read the stop codon. We have this release factor or termination factor that comes in and binds here that basically stops the process. And so now we stop the creation of our protein. And so now we're left with this series or this chain of amino acids. Okay? So as far as today's attendance code word, let's use translation as the code word for today. Okay. So today's code word for attendance is translation.